So guys, the first enzyme is DNA helicase, our unzipping enzyme. Helicase will separate double-stranded DNA into single strand, allowing each strand to be copied. During DNA replication, helicase will unwind DNA at a position called origin, where synthesis will be initiated. Helicase continue to unwind the DNA, forming a structure called the replication fork, which is named for the fork appearance of the two strands of DNA as they are unzipped apart.
After that, proteins called single-strand binding proteins (SSBP) create the separated strands of DNA near the replication fork, keeping them from coming back together into a double helix. As well as stabilizing the single-stranded DNA, SSB proteins bind to and modulate the function of numerous proteins involved in all of these processes.
Next is primase, the initializer enzyme. With as great as DNA polymerase is, poor DNA polymerase cannot figure out where to get started without something called primer. Primer is a short RNA sequences that are complementary to a single-stranded piece of DNA, which serve as its template. It is important that primers are synthesized by primase before DNA replication can occur. This is because the enzymes that synthesize DNA, which are called DNA polymerase, can only attach new DNA nucleotides to an existing strand of nucleotides.
Hi guys, so we're going to talk about DNA polymerase III. DNA polymerase III is the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. It can only work the 5' to 3' direction because it can only add nucleotides to the 3' end. DNA polymerase III has a high processivity and therefore synthesizes DNA very quickly. The high processivity is due in part to the beta clamps that hold onto the DNA strand. Okay, now DNA polymerase can get to work. Remember, it is an important enzyme that adds DNA bases. DNA polymerase III activity begins after strand separation at the origin of replication. As replication progressed, DNA polymerase III arrives at the RNA primer and begins replicating the DNA by adding onto the 3' OH of the primer. DNA polymerase III will then synthesize a continuous or discontinuous strand on DNA depending if this is occurring on the leading or lagging strand.
Did you know the physiological function of polymerase I is mainly to repair any damage with DNA, but it also serves to connect Okazaki fragment by deleting RNA primers and replacing the strand with DNA.
Here comes enzyme ligase, the gluer. Ligase fills in missing nucleotides on the lagging strand of DNA. Since the leading strand is being read and added to in the same direction as the helicase complex, there are no gaps.
DNA nucleases catalyze the cleavage of phosphodiester bonds. These enzymes play crucial roles in various DNA repair processes, which involve DNA replication, base addition repair, nucleotide addition repair, mismatch repair, and double-strand break.
Topoisomerase I enzyme cut a single strand of the double helix past the other strand through the cut and reseal the break, producing the overwound molecule, which now has one favorite twist. Topoisomerase II enzyme do the same thing but with both strands of the double helix. Topoisomerase II cuts both strands of a double-stranded DNA and passes another double strand through the break and then reseal the break.
Telomeres is the region of the end of chromosome. Telomerase is an enzyme that extends the telomeres of chromosome. It is RNA-dependent DNA polymerase, which means that it is an enzyme that can make DNA using RNA as a template. The enzyme binds to a special RNA molecule that contains a sequence complementary to the telomeric repeat. It extends the overhanging strands of the telomeres DNA using this complementary RNA as a template. When the overhang is long enough, a matching strand can be made by the normal DNA replication machinery, produce double-stranded DNA.